Hello, welcome to my presentation. My name is Qingxin. I'm from SFU Biogates Research Group. Then I'm going to talk about my study, electrochemical pressure impedance spectroscopy as a diagnostic method from polymer nitride fuel cells. So we're going to go through a brief introduction and how it developed them based on a classical diagnostic method, electrochemical impedance spectroscopy, and how it's been implemented onto a test station and go through results sections, flow channel, and electrochemical response. And then we'll go to summary and conclusion. So the need of diagnostic tools are increasing nowadays. We have 40,000 fuel cell power vehicles now, and becoming more and more fuel cell power stuff in buildings, transportation industry, and diagnostic tools are essential in fuel cell life cycles, including manufacturing, operation, and maintenance. So in the fuel cell, we have a hydrogen splitting to protons and at the anode catalyst layer carried by the polymer nitride membrane to the cathode, becoming the water at the cathode. And there is a current flow through from the anode to cathode as in the form of a proton. And if we measure the response between the current and voltage, we can get all these diagnostic parameters within these current charge transfer processes, including the catalyst layer and the polymer nitride membrane. However, and many transport phenomenon doesn't involve a charge transfer, including for example, the oxygen diffusion and the water removal. Especially at the cathode side, the mechanism more complex and there are more factors involving. So in order to do this, if we can use the pressure as a perturbation source and measure the voltage accordingly, we're able to get these parameters about the cathode flow field and cathode diffusion layer and the diffusion inside the cathode layer. Luckily, if we get the pressure difference between the flow field inlet and outlet, and then measure the response between two different pressures, we can get this characterize the flow field alone only by these two different pressure profiles. So the objective of this research is to develop an EPIS method for diagnostic purposes, and it should be easy to implement onto an existing test station, and has a frequency range from, like, for example, like three order of magnitude. And the first prerequisite is to categorize the flow channel to know what's really happening inside the flow field plates, and get the determinant factor. How do we get a diagnostically favored API signal, including load, oxygen concentration, and flow rate? And based on that, we'll suggest what kind of parameters for this type of diagnostic tool can probe. So the implementation is based on this is on a bed pressure valve used to regulate the pressure of the whole fuel cell station, and not so now if once we can fix the whole fuel cell test station cathode at a certain pressure. So this back pressure controller controls through a pressure transducer right before this back pressure controller, and by reading this back pressure controller, reading this pressure transducer next to the back pressure controller, I can adjusting this pressure and give a sinusoidal wave pressure, and therefore we can create a pressure perturbation by controlling the pressure by this back pressure controller. So we have two different test cells. One has a serpentine flow channel, which has a 50 centimeter square of MEA. I use a naphthalene as a membrane and use 0.3 milligram per centimeter square platinum as a catalyst. And the other one has a different type of cell design. It has a parallel flow channel, and it also has a smaller cell area as 12 centimeters square. So the experiment were carried on 70 degrees C, and the normal operating temperature of for a fuel cell, and also the anode and cathode was humidified to like 95% RH, and both anode cathode before we started any test, the pressure was fixed at 100 kPa at the inlet, and the pressure oscillation amplitude was 1 kilopascal. So if we not withdrawing any current from the fuel cell and only pass through a pressure perturbation signal from at the outlet, what we saw at the inlet, actually there is a phase difference between two pressure signals, and the inlet pressure signal amplitude is much smaller than the outlet pressure signal amplitude. This is because the flow channel actually act as a low pass filter, and the flow channel contains a resistant part, actually causing this flow pressure difference between the inlet and the outlet, also a capacitance part to store gas. So if we scanning through a range of frequencies and fit all this data into a low pass filter model like the RNC, and we can get uh, this flow channel resistance and capacitance based on these parameters, and we can also find the characteristic frequency of this flow channel uh, because I also call cutoff frequency. If the pressure signal is lower than this cutoff frequency and the amplitude is doesn't change and their phase is on shifted almost zero. If the pressure frequency is higher than this cutoff frequency, and the amplitude of the inlet pressure oscillation dramatically decrease and eventually goes to zero. And therefore, in the results, we can see the flow channel resistance increasing even with increasing flow rates, and the capacitance, especially at the second cell, is almost a constant. And we can, from all these different flow rates, we can get the characteristic frequency. So we're able to know the operating range for frequencies for VPIS. If we know like at what range like the pressure is not affected by the flow channel. So for a typical EPS response, we'll see this voltage oscillation, and the voltage oscillation contains a phase difference between the voltage and the pressure, and we assuming once we have an EPS response, if we increase the oxygen stoichiometry by decreasing the load, eventually the EPS response will diminish. And if we bring back the oxygen stoichiometry to a lower value, for example, bring down the oxygen concentration by mixing with nitrogen, or bring down the flow rates, and this is not applicable for a second fuel cell testing cell, and it will eventually bring back the EPS response. But bringing up the pressure oscillation amplitude doesn't help because it's a response between the pressure and the voltage. And so therefore, we can see this results basically follows the it basically follows the assumption. If we bring down the current density for both testing cell, and we can see this EPI's Nyquist circle become smaller and smaller, and eventually gonna become like not diagnosed favor because it's so small and the voltage response is buried in the noise. And if we at this very end with low current densities, if we bring down the oxygen concentration, we can see this EPI's curve, Nyquist semicircle becoming larger and larger. Eventually, we bring down this oxygen stoichiometry to where it was, like for 2 and 3, for low value, we're able to get the CPI's response back. So same observation was found to bring down the flow rates, and actually at lower flow rate, we can see this EPI response recovered. So in summary, you have characterized both flow channels and also find the resistant capacitance of these two flow channels and implement this type of EPS or back pressure valve and to an actual test station. And overall, the signal depends on certain oxygen depletion. We need for if you want a diagnostic favored EPS response, we will need a lower oxygen stoichiometry ratio. Higher oxygen stoichiometry ratio, which means like oxygen is more easy to that diffuse to the active site, actually is which means like has a smaller impedance, and the EPS the response signal becomes smaller and smaller, eventually diminish, and we will not able to get any diagnostic favored signals. So at last, I would like to thank Dr. Byron Gates, Dr. Michael Ikering, and my supervisory committee members, Stephen Hawcroft and Gary Leach, and all my group members here, and all the support from Greenland Innovation based on their test station and their techniques. So thank you very much. So here is my presentation.